Hello guys, welcome back to my YouTube channel. This lecture is about cerebral cortex, and we're gonna do it based on three separate headings: structure, efferent inputs, and neural circuits. I've already made a video about the physiological anatomy, or basically just gist of the anatomy of the cerebellum in a separate video, and now we go into the cerebellar cortex. So obviously the cerebellum will be made up of white matter and gray matter. The gray matter is in the periphery formed by the cerebellar cortex, whereas the dark matter, it seems, white matter is more central, and it consists of nuclei. So which nuclei? Here we have four: doctors eat good food, dentate nucleus, emboliform nucleus, globus nucleus, and fastigial nucleus or nucleus fastigi. So that is the white matter, whereas in the gray matter we see three layers: the inner granular layer, the middle Purkinje cell layer, and the outer molecular layer. Molecular layer consists of nerve endings and nerve cells. So which nerve endings does it consist of? We can see clearly in this layer, in this picture. Here we have the white matter, the granule cell layer, this is the Purkinje cell layer, and then this is the molecular layer. So we can clearly see the dendrites of the Purkinje cell layer are there as nerve endings in the molecular layer. Then this is a granule cell whose dendrites are here in the granular layer, whereas the axons are extending into the molecular layer. So the second is the axons of granule cells. The third we can see are the climbing fibers. You can see the climbing fibers, which are the efferent fibers, are going and they are synapsing with the dendrites of the Purkinje cells. So even climbing fibers are present as nerve endings in the molecular layer. What are the nerve cells present? We have two. We have the basket cells and we have the stellate cells. So these basket cells and stellate cells have their own function, which we'll talk about later in this video, but these are also present in the molecular layer. Next is the Purkinje cell layer. So these layers of the cerebellar cortex we have seen in the histology slides also. So many times we have discussed about the three layers, and the most distinguishing feature about that particular slide is the Purkinje cell layer, because the Purkinje cell layer is only one cell layer thing, which is only made up of Purkinje cells. So this is the middle Purkinje cell layer consisting of the cell bodies of the Purkinje cells. Then we have the granule cell layer, which contains two types of cells: the granule cells and the Golgi cells, which you can see here again. We already talked about the granule cells, and here are the Golgi cells, and it will be very interesting to know how all of these interact with each other. Here also you can see the layers. This is from the A.K. Jain textbook. You have the white matter here, from where the climbing fibers are coming and they're going there, and they're synapsing with the dendrites of the Purkinje cells. This is the one Purkinje cell. These are all the Purkinje cells in the Purkinje cell layer. You can see how profusely the dendrites are being formed. Stellate cell and basket cells in the molecular layer. Golgi cell, more specifically here they have shown it in the middle Purkinje cell layer, but it's there in the granule cell layer, and we'll go into the details of all of this. So that was about the structure, the three layers. Now coming to the efferent inputs. So we have the climbing fibers and we have the mossy fibers. So climbing fibers and mossy fibers are the efferent fibers. That means they bring the connections or efferent inputs from other parts of the body to the cerebellum, or more specifically to the deep nuclear cells present in the white matter, which gives out the output. So they do it either directly by giving fibers to the deep cerebral nuclei, or they do it through the Purkinje cells. So when they do it directly, it's causing a positive, it's exciting the deep nuclear cells, whereas the Purkinje cells are inhibiting the firing of the deep nuclear cells. So both of these are happening simultaneously 24/7, but normally it is in the favor of excitation. So the climbing fibers, as you can see, they are coming from the white matter, then they are going through the the molecular granule cell layers, and they are reaching the molecular layer. They are synapsing with the dendrites. We already talked about this. So what is there to discuss about this is that they are coming from the inferior olivary nucleus. We are getting the climbing fibers. So one thing they're doing is they're doing an excitatory effect on the deep nuclear cells. The second thing is it is going and anastomosing with the Purkinje cell dendrites that we discussed about. One climbing fiber is supplying at least, or innervating, you could say, or synapsing with 10 Purkinje cells, and there are 300 synapses with each dendrite of the Purkinje cell. So you see how only if one climbing fiber is excited, the Purkinje cell will be so hugely affected or excited, or the synapse will take place, the action potential will go, and this potential is a single prolonged peculiar action potential, which is known as a complex spike. It is long, the duration of it is long, it's a very strong spike. There is a primary strong spike, and then eventually the spikes get weaker. That's why the primary strong spike is followed by a trail of weakening secondary spikes. So that is how the action potential of the climbing fibers is there to the Purkinje cells. Apart from that, it is giving collaterals to the Golgi cell bodies also, which are there in the granule cell layer. So the climbing fibers, they come from the white matter, right? They supply the deep cerebellar nuclei, then they go from there, the dendrites of the Purkinje cells, and then they are collaterals to the Golgi cells also. So here you can see from the climbing fiber supplying the deep cerebellar nuclei, then going supplying the dendrites, and then there will be a collateral to the Golgi cell. Done. Now we come to the second type of afferent fibers, which are mossy fibers. These are coming from the brainstem, spinal cord. So you can say the spinocerebellar tracts, the pontocerebellar tracts, cerebellocerebellar tracts. So that is what the mossy fibers are about. So the mossy fibers go and they synapse with the granule cells. More clearly the dendrites of the granule cells. So those granule cells, then their axons, they are forming the parallel fibers. So mossy fibers are directly not supplying the Purkinje cells, they are doing it through granule cells. So the mossy fibers synapse with the dendrites of the granule cells, which give parallel fibers. These parallel fibers go to the Purkinje cells. So in this case, 50 to 100 granule cells are going and reaching one Purkinje cells. That means you need to excite a large amount of granule cells to excite one Purkinje cell. Otherwise, it will be not effective. So for this particular circuit to work, it will need time, because it will need to excite so many cells. So for the potential to build up, in order for the Purkinje cell to get the potential or fire, we need some time, and because it requires so many cells. So that is how mossy fibers work. These are weaker, short duration potentials. That's why it is called as the simple spike, whereas here it was called as the complex spike. So that's how the mossy fibers are working. Now we have the third type of, not the third type, basically there are only two afferent fibers, climbing and mossy. So the third topic for this lecture was neural circuits. So we have ended up actually talking about them already. This is a circuit, this is a circuit. So what does Purkinje cell do? It inhibits the deep nuclear cell, right? So we talked about that. Now we'll talk about the stellate and basket cells, which I have talked to you about. So we talk about two types of basically things that happen in this circuit, that are the feed forward inhibition and the negative feedback loop. That is what is left all over. So talking about the stellate basket cells, the granule cells, or you could say the mossy fibers, or you could say the parallel fibers, they also synapse with the stellate and basket cells. These stellate and basket cells, they release IPSPs, or basically inhibit the Purkinje cells. So you see some fibers from the parallel fibers, some of the parallel fibers, they're actually exciting the Purkinje cells, whereas the ones that are coming from the stellate and basket cells are inhibiting the Purkinje cells, right? So if these are the parallel fibers supplying the stellate and basket cells, the axons will be from the stellate and basket cells will be at 90 degrees to the parallel fibers. So that is also one of the point. So you can see here the mossy fibers synapsing with the granule cells, producing parallel fibers. It is exciting the Purkinje cells. It is also supplying the stellate and basket cells, which is doing lateral inhibition of the Purkinje cells. This kind of inhibition is called as the feed forward inhibition. It helps to limit the duration of the excitation here. Alright, that is one thing, and it is also called as lateral inhibition. Why? Because how lateral inhibition works, it sharpens the potential. So that's also how it works. So this is the feed forward inhibition by the stellate and basket cells. So stellate and basket cells are producing inhibitory effect on the Purkinje cell. Until now we saw climbing fibers, mossy fibers, granule cells, all these were doing excitatory effect on Purkinje cells. The first we saw are the stellate and basket cells, which are doing inhibitory effect. Now we talk about the negative feedback loop. The negative feedback loop comprises of the Golgi cells. So Golgi cells are present in the granule cell layer. So we see here that the dendrites of the Golgi cells are present in the molecular layer, and then we have the cell bodies here in the granule layer. So this cell bodies will also receive some inputs, and the dendrites will I'm totally wrong. These are the axon. I think these are the dendrites. So what I was trying to say is, the cell body and the dendrites, they both receive separate inputs. So as I said, the climbing fibers, they send collaterals to the Golgi cells, to their cell bodies. So here we can't see it in any of the diagrams. Yes, but yeah, so the climbing fibers, they send, and then Purkinje cells also send these certain fibers to the cell bodies of the Golgi cells. Apart from that, the granule cells, which gives parallel fibers, these parallel fibers are also going and synapsing with the dendrites of the Golgi cells. So we have the climbing fibers and the Purkinje cell fibers, which are synapsing with the cell body, and then we have the dendrites, which are getting it from the parallel fibers of the granule cells. So because of this effect, the Golgi cells, they make inhibitory synaptic connections with the dendrites of the granule cells itself. Granule cells से आ रहा है, granule cells को inhibit कर रहा है. So because it is making inhibitory connections with the dendrites of the granule cells, what are there at the dendrites of the granule cells? Mossy fibers, right? So because of these inhibitory connection, the transmission from the mossy fibers to the granule cells is inhibited. So the Golgi cells, because of its inhibitory synaptic connections with the dendrites of the granule cells, inhibits the transmission from the mossy fibers to the granule cells. So this is basically inhibited. So this is called as a negative feedback loop, because this is getting excited because of granule cells, and this itself is inhibiting the granule cells. So this is a negative feedback loop, which I wanted to talk about. Low power mode, it seems. Okay, so the video is gonna end soon, definitely. So one more thing I had to talk about in the neural circuits is the turn on turn off output signals from the cerebellum. So in this case we talk about mossy fibers. All right, so whenever we're doing a muscle movement or we are learning a new movement or anything, we need the agonist muscles to turn on and the antagonist muscles to turn off so that we can start the movement. And towards the end of the movement we need the antagonist muscles to turn on and the agonist muscles to turn off. So this is how does it happen in the cerebellum is what we are gonna discuss very quickly. So whenever there is a signal from the cerebral cortex, okay, we have to make this movement, so there is one pathway which directly goes to the muscle. Okay, it directly goes to the muscle to begin the initial contraction, but at the same time there are parallel signals which are going to the cerebellum by the way of the pontile mossy fibers. So this one branch of the mossy fibers goes directly to the deep nuclear cells which are in the dentate or in the other deep cerebellar nuclei. So this sends an excitatory output into any of the output tracts of the cerebellum. For example, the cerebral corticospinal motor system. So through the thalamus or whatever, however it is, through the thalamus to the cerebral cortex or by way of neuronal circuitry in the brainstem, whatever it does, this is how it supports that contraction of the muscle. This is how the turn on signal is put. So after a few milliseconds, the initial signal which went to the muscle, it becomes even stronger. So when the cerebellum is intact, the normal function is going on, the normal effect is going on. But just in case there is an absence of the cerebellum, the secondary extra supportive signal is missing. So in cerebellar, support makes the turn on muscle contraction much stronger than it would be if the cerebellum was not there. Now the second thing is the turn off. What causes the turn off signal for the agonist muscle when we have to terminate the movement? That is where the Purkinje cell comes. So the same mossy fibers which are giving the deep cerebral nuclei, they are sending impulses to the granule cell, to the parallel fibers, to the Purkinje cells, and the Purkinje cells are inhibiting these deep nuclear cells from firing. So the Purkinje cells in turn inhibit the deep nuclear cells. So this pathway is, why is it happening? It's happening in the same time, but why is that is happened later? Because of the slower spikes, the weaker short duration simple spikes, and the slowest conducting fibers. The parallel fibers are the slowest conducting nerve fibers in the nervous system, which are there in the cerebellar cortical molecular layer. So these fibers are weak, the signals are also weak, as I said, simple squal. So they, as I said, they require some time to build up to excite the Purkinje cell. So once it is excited, it sends a strong inhibitory signal to the same deep nuclear cell, and the signal helps turn off the movement. So did you see how this is precisely timed? When we start a movement or we end a movement, the turn on and turn off happens. This is for the agonist muscles, and a similar kind of stuff also happens with the antagonist muscles, but we don't need to go into the details as of now. We only talk about the agonist muscles for this discussion. So it is so precisely timed. If it wasn't timed, if the turn off didn't happen at the correct time, it will lead to oscillatory movements that will be discussed in the lesions of the cerebellum. So how does the cerebellum do this? How does it time it so perfectly? It is because the Purkinje cells, they learn to correct the motor errors. That means the first time you do any movement, when you learn a new movement, are you very good at it? You are not. You are flimsy with that movement. But the more times you do it, the more times the Purkinje cell learns to correct the errors that are being happening in your movement, and that's how you eventually are skilled in doing that movement, and then the turn on and the turn off happens at the correct time. So that's how the neural circuitry works. That's how the functional unit of cerebral cortex, that is basically this is the functional unit of the cerebellum. That's how it works. Okay, so that's all for this video. I hope you understood what I was trying to say, and I'll see you guys next time. Bye.